Okay, so again, good morning. For today, we will be discussing about the cytokines. So perhaps during the past year, you have been hearing about cytokine storm being brought about by severe COVID cases. So what are these cytokines? Cytokines are actually small proteins. So when you say small proteins, they are actually proteins of very low molecular weight. Okay, and cytokines have um, important roles because they can, number one, control the growth, and another, growth and activity. So they can control the growth and activity of the immune system, and also they can control the activity and growth of our blood cells. Meaning to say, they are also important for our hematopoiesis. So cytokines are important, okay? And and whenever they are released, so cytokines will be released, okay? So they will actually go to the effector cells. So whenever these cytokines will be released, they will go to the effector cells. So what do you mean by effector cells? Effector cells are like target cells, meaning to say, um, the action of the cytokines will be take in the effect of the cytokine actions will be seen in the effector cells. Okay, so they can affect the growth not just of the cells of the immune system, but cytokines can also affect the growth of the blood cells. Okay, so meaning to say, um, whenever there is an abnormality or an ideal situation in our body, so cytokines activity, of course, is manifested during infection and inflammation. Okay. Is it a good thing for our body? Yes, because it will actually help our body to mount an efficient immune response against pathogens. But is it also bad at the same time? Yes, because during severe infection, our body becomes the battleground. Our body becomes a war zone, and too much cytokines will also damage our own cells. That's the reason why some of the patients with severe COVID-19 are actually severely affected as well by the cytokine storm. So when you say cytokine storm, each cells that are capable of producing cytokines are actually are actually being produced all at the same time. So it's literally a storm. Okay, so that is the reason why for today we will try to, of course, discuss cytokines, and these are the these are the these are the outline. So I have already introduced cytokines, and then cytokines are involved in both innate and adaptive immune response. So we will be discussing them. And then the Th17 cytokines, the hematopoietic growth factor, the cytokines and anti-cytokine therapies, and clinical assays for cytokines. Okay. So as what I told you a while ago, cytokines are small, soluble proteins. Okay. The word soluble is very important. Okay. So the reason why they have to be soluble is that so that they can efficient in affecting or in regulating the effector cells. These effector cells may be directly involved in both innate immunity or adaptive immunity. Okay, so when you say adaptive immunity, this is the adaptive response of our immune system towards various kinds of infection. So cytokines, meaning cytokines are actually a chemical substance being produced by a cell. So it's actually the cyto here refers to cells. So if you hear the term lymphokines, the cells are very specific. Now we're talking about lymphocytes. Or if you've heard the word monokines, then the cytokines are actually have actually originated from monocytes. So, but a more general term, of course, would be cytokines. And these particular small soluble proteins are being produced whenever there are stimuli. So there are various chemical stimuli that would initiate the production of cytokines. So to name a few, we have bacterial lipopolysaccharide. So this is found in the cell wall, okay, bacterial lipopolysaccharide cell wall of gram-negative bacteria. Flagellin is, of course, obviously, is the protein, the flagella. So other bacterial products and the ligation of cell adhesion molecules. Okay, so ligation of cell adhesion, because the ligation here represents tissue injury. So whenever there is a tissue injury, so cytokines will also be produced. So these are just some of the few stimuli that would actually induce this production of cytokines. So cytokines act differently. Okay, so they if together they act as one, so we call it synergy. Okay. Now sometimes some cytokines have this tendency to multitask, meaning to say one cytokine has many actions. So if that's the case, we use the term pleiotropy when one cytokine has many actions. Okay. Now, when you say redundancy, it means that different cytokines have the same function. Okay? And antagonism, excuse me, antagonism is when cytokines counteract each other. So antagonism is the exact opposite of synergy. Because synergy means that they act together, but when you say antagonism, they counteract each other. To give you an idea, I would like to present to you a graph that would summarize the function. So let's say, for example, this is cytokine A alone, and then this is cytokine B alone. Okay? Cytokine A and cytokine B, when you combine them, okay, the function of cytokine B increases. Meaning to say, cytokine A synergizes with cytokine B. Okay. Now, this is the target cell response when cytokine C is alone. So when cytokine A and cytokine C combine, the target cell response of cytokine C drop significantly. So meaning to say, they counteract each other. So in this particular graph, we could say that cytokine A antagonizes with cytokine C. Okay, so that's the difference between synergism, and antagonism. Okay, so let's talk about the in vivo effects of cytokines expression. Okay, so when you say in vivo effects, these particular effects are actually observed, okay, in the body, inside our body. Okay, so there are in vivo effects of cytokines expression on leukocytes and other cells. So number one is regulation of growth. Okay, so regulation of growth. So it affects, of course, the size, and of course, growth doesn't just pertain to size. Okay, it also pertains to functions. The same thing with differentiation. Differentiation refers to maturation from being a blast cell to a fully mature cell. Okay, so this is differentiation. And the third effect of cytokines is gene expression. Okay, so when you say gene expression, there is already an observable effects due to the presence of a certain gene. So that is gene expression. Now, cytokines will also affect the expression of leukocytes when it comes to gene expression. So these are the different in vivo effects of cytokines on leukocytes, not just on leukocytes, but other cells as well. Okay, cytokines are actually produced through the following, no? So we have autocrine stimulation. So when you say autocrine stimulation, this also refers to self-stimulation. Okay, so for example, antigen-presenting cells produces interleukin-1 beta, and interleukin-1 beta will make the antigen-presenting cells such as this macrophage to produce more interleukin-1 beta. So this is an example of autocrine stimulation. Okay, so one cell will stimulate itself. Okay, so when you say paracrine stimulation, the effects is actually geared toward the nearby cells. So kailangan malapit lang. So for example, um, interleukin-1 beta will affect Th2 upon binding. Di ba? If you remember, Th2 has CD4. Th2 has CD4, while the antigen presenting cell has MHC class 2. So upon binding, interleukin-1 beta affects the functions of T helper cell 2 because there's a binding, or in this case, so this is an example of a neutrophil. There is no binding, but at least this neutrophil and antigen presenting cell is nearby. Okay, so that's what we meant by paracrine stimulation. When we say systemic or endocrine activities, though it's occasional, but the effect is actually geared toward, okay, a distant effector cell. Ibig sabihin, malayo yung effector cell. So that's the reason why it's systemic. So, for example, it needs to travel through the blood vessel. So interleukin-1 beta would travel, since it's soluble, it's capable of traveling within the blood vessel, would eventually affect the liver cell. And in the liver cell, we have the Kupffer cells, okay, that could be activated as well. Or it can even affect the brain. And then the brain uh, would have an effect on the dendritic cell, for example. So, even if it's distant, the effect is still possible. So if that is the case, we call it the systemic or the endocrine effects of the cytokines. Okay, so I hope that's clear. So we have the autocrine, paracrine, and systemic or the endocrine. Okay, so a cytokines would also have multiple actions. Okay, and multiple actions are due to the reason that there are various receptors on many cell types. Okay, so if you have the receptors for a particular cytokines, if a certain cell would have a receptor for a particular cytokine, then that cell, okay, will be considered as an effector cell because they have the receptor. And also, cytokines would also have the ability to alter expression of numerous genes. Okay, so meaning to say, I've been telling you this a while ago, is that it affects gene expression. Okay, so cytokines may share properties and receptors, and they may have an overlapping effect. Okay, so that's the reason why we have the so-called pleiotropy, redundancy, because of the fact that there are multiple receptors, they share properties, and they have overlapping effect. So cytokines can determine whether the host will be able to mount an effective defense or not. Okay? And that is also the reason why, according to the previous studies, children do not have severe, it's very rare for children to have severe COVID as compared to adult, particularly because of the fact that they have the cells of their immune systems are not yet fully developed and minimal cytokines are being produced. So, they do not really mount a defense, an exaggerated defense against pathogens. Unlike us, our immune cells are fully developed. So when we mount an immune response against a certain pathogen, we mount it in such a way that we produce cytokines exaggeratedly. And if that's the case, then our own cells will also be affected. So these are the major cytokine families. So we have the tumor necrosis factors. We have the interferons. We also have the chemokines. And then we also have the transforming growth factors. And then we do also have the colony stimulating factors. Okay, so these are the different classifications of cytokine families. Okay. Now, in today's discussion, we will be dividing our discussion into two major topics. So we will be discussing first the cytokines that are involved in the innate immune response. Okay? So when you say innate immune response, these are the ones that are involved in natural immunity. Meaning to say, these cytokines are produced, okay, in response to a non-specific pathogen. So that's the reason why natural immunity is also known as non-specific immunity. Okay. So there are cytokines that are involved in triggering the recruitment of effector cells. So to name a few, we have interleukin-1, tumor necrosis factor alpha, interleukin-6, chemokines, transforming growth factor beta, and alpha and beta interferons. Okay, so these are the different cytokines involved in triggering the recruitment of effector cells. Okay. So let's talk about interleukin 1 first. Okay. Interleukin 1 is an endogenous pyrogen. Okay. So when we say pyrogen, it induces fever in the acute phase response. This is the reason why whenever we have infection, our temperature increases because interleukin 1 will exert its action on hypothalamus, and our hypothalamus controls our body temperature. Okay, so that is the reason why having a fever is actually a good indicator that you have an infection. And that is the reason why whenever we enter a mall and other public places, they are actually checking our body temperature to check if we have an infection or not. And so interleukin 1 is the reason for the presence of fever. So interleukin 1 induces the production of vascular cell adhesion molecules. It will also act, okay, to produce other cytokines. So these are the other cytokines that will be produced, okay, that will be as a result of interleukin 1 action, no? So chemokines, interleukin 6. Okay, so interleukin 1 is also divided into several subcategories. So we have interleukin 1 alpha, beta, and RA. Okay. So interleukin 1 alpha and beta are pro-inflammatory cytokines, and they are being produced by monocytes, macrophages, and dendritic cells. So again, these cells are examples of antigen-presenting cells. So it is the antigen-presenting cells such as the monocytes, macrophages, and dendritic cells that would produce interleukin 1 alpha and interleukin 1 beta. Interleukin 1 beta is responsible for most of the systemic activity that is attributed to interleukin 1. So between A and B, between alpha and beta, it's more of the alpha. Okay? So they are important in fever, the reason why there is a fever. Okay? Activation of phagocytes. So phagocytes are cells that are capable of phagocytosis. Mostly these cells are neutrophils and monocytes. And then also they are involved in the production of acute phase proteins. Well, we have already discussed that the most common example of the acute phase protein is the C-reactive protein. Interleukin RA is produced by monocytes and macrophages. So RA, it acts as an antagonist to interleukin 1 by blocking the interleukin 1 receptor and limiting the availability of the receptor for interleukin 1. So it's a regulatory antagonist. Okay? So it regulates interleukin 1. So we do not want to produce too many interleukin 1 because if interleukin 1 beta is being produced in enormous amount, imagine we will be constantly in high fever. We will be constantly inflamed due to the production of enormous amount of acute phase proteins. So to regulate such an event, then this is where interleukin 1 RA will come into play because they will antagonize the action of interleukin 1 alpha and interleukin 1 beta. So that's the function of interleukin 1 RA. Okay, so let's talk about the tumor necrosis factor family. So among the family of tumor necrosis factor, the most prominent of them all is the alpha. And it exists in two forms, membrane bound, and the other one is soluble form. Okay? So when you say membrane bound, it means that they are attached to a certain cell membrane. And then the soluble form means that soluble form means that they are free. Okay? So they can cause vasodilation and increase vasopermeability. Okay? So vasodilation means that the lumen of the blood vessel expands or widens. And because of that, okay, fluid will come inside the blood vessel. So there is an increased vasopermeability. And too much vasopermeability will cause edema and eventually swelling. Okay. However, vasodilation is also important because this will allow diapedesis to be more efficient because the spaces in between endothelial cells increases as well. So it is being triggered by the presence of gram-negative bacteria. Because in gram-negative bacteria, the cell wall has an LPS, lipopolysaccharide. So LPS, therefore, is actually an endotoxin for gram-negative bacteria, and they are a very good antigen. Good enough to allow alpha tumor necrosis factor to be released. Okay, so there you have it, the alpha tumor necrosis factor. Okay, so again, it is being secreted by activated monocytes and macrophages. The alpha tumor necrosis factor can also activate T cells through its ability to induce expression of MHC class II molecules. Remember, MHC class II molecules are found in antigen presenting cells such as macrophages. Okay, so alpha tumor necrosis factor somehow would have a similar action to that of interleukin-1 because of its ability to activate T cells to induce expression of MHC class II molecules. Okay, so that's alpha tumor necrosis factor. Again, it's still involved in innate immune response. So let's talk about interleukin-6. Interleukin-6 is a single protein produced by lymphoid and non-lymphoid cells. So it can either be produced by lymphoid and non-lymphoid cell. So it is a part of cytokine cascade release in response to lipopolysaccharide. Again, found in the cell wall of gram-negative bacteria. Okay, it is a pleiotropic. So again, when you say pleiotropic, it has several functions, it has several actions and play a role in acute phase reaction. So interleukin-6 is being triggered by interleukin-1. Okay. So interleukin-6 stimulates B cells to proliferate and differentiate into plasma cells. Okay, so B cells will eventually become plasma cells. Why? Because plasma cells will eventually secrete antibodies. Therefore, there will be what? Humoral immune response. Take note, guys. Whenever there is a secretion of antibodies, we call that a humoral immune response. Interleukin-6 will also induce CD4 positive T cells. Ano yung CD4 positive T cells? These are T helper cells. Okay? So interleukin-6 will induce CD4 positive T cells to produce greater quantities of both pro and anti-inflammatory cytokines. So you might be wondering why pro-inflammatory cytokines are being produced at the same time with that of the anti-inflammatory cytokines. Because this will regulate inflammatory response. You do not want to have too many pro-inflammatory cytokines because that will be difficult, okay, for the patient. So in response to the production of pro-inflammatory, anti-inflammatory cytokines would also be necessary so that it will not be the person who is infected will not have that too much burden of having so many pro-inflammatory cytokines. Okay, so let's talk about the chemokines. Chemokines will enhance the motility and promote migration of many types of WBC towards a certain chemokine. So we call it chemotaxis. Now, to simply understand chemotaxis, so remember that whenever we say chemotaxis, we are referring to chemical attractions. So whenever chemokines are being stimulated by chemical, or whenever whenever WBC are being stimulated by a certain chemical, we call that chemotaxis. Okay? So chemokines will enhance the motility and promote the migration of WBC. So WBC will travel faster because of the chemokines. And we are classifying chemokines into 4 families based on the position of the N-terminal cysteine residues. Okay? So if it's located in between a certain amino acid, if the cysteine amino acid is located in a certain amino acid, we call it alpha. But if the cysteine amino acid is together and there's no amino acid in between, we call it beta. But if there's only one cysteine, we call it C. And CX3C means that there are 3 amino acids in between 2 cysteine molecules or 2 cysteine amino acid. There are still 3 amino acid. So CX3C. Okay, so these are the different classification of the chemokines. Chemokines are involved in the initiation and development of inflammatory response in numerous disease processes. So it combined with cell adhesion molecules, facilitate the extravasation of leukocytes. Again, extravasation of leukocyte means that WBC goes out of the blood vessel. So this term is simply put, we call it diapedesis. So WBC will go to the area of inflammation because of the chemokines. So that is the reason why there would be what? Abscess or nana. And the reason why WBC goes into the site of inflammation, one of the factor is chemokines. Okay. So another family involved in the innate immune response, this is the transforming growth factor beta. And there are 3 isoforms. We have the TGF beta 1, TGF beta 2, and TGF beta 3. So it induces anti-proliferative activity. This one is very important. Anti-proliferative activity in many cell types. So when you say anti-proliferative, it opposes or it prevents differentiation. Okay? And growth. It's very important, particularly if the effector cells are tumor cells. Because we do not want tumor cells to proliferate excessively or uncontrollably, because this will result to metastasis and eventually death of the person. So it is the transforming growth factor beta that will regulate the cell growth, differentiation, apoptosis or the programmed cell death, migration, and inflammatory response. So it helps down-regulate the inflammatory response when it is no longer needed. If there are no more stimuli, if there are no more pathogens, we do not want inflammatory response. We do not want to be in pain forever. So it is the transforming growth factor beta that would down-regulate such an event. So the transforming growth factor beta will inhibit the activation of macrophages and the growth of different somatic cell types. So it functions as an anti-inflammatory factor for mature T cells. And it also in activated B cells, it inhibits proliferation, because you do not want B cells to proliferate when they are no longer needed, particularly if there's no more pathogen. So we do not need too much proliferation of B cells. So it may function as an autocrine regulator to limit the expansion of activated cells. Okay? So kumbaga guys, basag trip to sa mga gina-gina immune cells natin na gustong-gusto nila mag-proliferate kahit wala na yung kalaban. Then, darating na si transforming growth factor beta to down-regulate such an event. Okay, so let's talk about the interferons. Okay, so the type 1 interferons, particularly the alpha interferons and the beta interferons. So they are antivirus or antiviral. Okay, so these particular interferons are being produced by the dendritic cells. So it induces the production of proteins and pathways that interfere with viral replications and cell division. So meaning to say, whenever the cell has interferon, whenever the cell has an interferon, then the neighboring cell will not be infected. So for example, this is the cell, and then this is another cell. A virus will enter this cell. So this cell is already infected. However, this particular cell that has been infected will produce interferon so that the virus will not be able to enter the cell. Okay, so because it interferes with viral replication. So the neighboring cell will be protected. Okay, so it has an antiviral activity. Not only that, interferons will also activate natural killer cells. And you know, natural killer cells can lyse target cells. So when we say target cells, this could be a virally infected cells. Type 1 interferon will also enhance the expression of MHC class 1 proteins. MHC class 1 proteins, they are found in all nucleated cells of our body. Therefore, type 1 interferons could be active in certain malignancies and other inflammatory processes. Okay, so this is another cytokines involved in innate immune response. Okay, so we have already discussed the cytokines involved in innate immune response. So To review, we have already discussed type 1 interferons, transforming growth factor beta, the chemokines family, interleukin 6, tumor necrosis factor alpha, and interleukin 1. Okay, so now let us discuss the cytokines involved in adaptive immune response. Okay, so adaptive immune response is actually produced in response to a specific antigen or specific pathogen. So that's the reason why the adaptive immune response is also known as specific immunity. Okay, so when the T cell receptor or the TCR on a CD4 positive T helper cell captures antigen, clonal expansion of the T cell occurs. Are you getting the first bullet? So for example, this is the T helper cells, okay? And the T helper cell has CD4, okay? And then this particular T helper cells, so for example, there is a macrophage here that presents an antigen to this particular T helper cell. Clonal expansion of T helper cell occur. And because of the clonal expansion, there are three possible types of T cells, which I have already discussed in the previous topic, and and this is part of the quiz. Okay, so we have the TH1, the TH2, and the Treg. Okay, so in order to become TH1, TH2, and Treg, so there are various cytokines that will be involved in this particular immune response. Okay, so this is a better version of the drawing a while ago. So for example, this is a macrophage, and then a naive CD4 means that it hasn't encountered any antigen. But since the macrophage presents the antigen, and this is the CD4, for example, this macrophage is no longer naive. Okay, so this macrophage is now, this CD4 is no longer naive. So upon presentation of antigen, this CD4 is now activated. And whenever the CD4 is activated, there will be what? Clonal expansion. Meaning to say, the activated CD4 could be TH1, Treg, or TH2. We will have a separate discussion for the TH17. Okay. Now, in order for the TH1 to be involved in cell-mediated immunity, these are the cytokines that are involved. In order for the TH2 to be involved in humoral immunity, these are the different cytokines that are involved. Treg would also have its own cytokine to upregulate or to downregulate the expression of the activated T cells. Okay, so that's what we meant by the different cytokines involved in adaptive immune response. So let us discuss them one by one. First, we will be discussing the TH1 cytokines. So what are the different cytokines involved in TH1? The first cytokine here is the gamma interferon. Okay. Do you know that the gamma interferon has the ability to affect the RNA expression level of more than 200 genes? Okay, so that's how influential, should I say, gamma interferon is. So it stimulates the antigen presentation by class 1 and class 2 MHC molecules. So take note ha, gamma interferon will not only affect class 1 but class 2 MHC molecules as well. So gamma interferon will activate macrophages to stimulate the phagocytic and cytotoxic abilities. Okay, so it will activate macrophages. It also regulates the action of CD4 Th cells, CD8 cytotoxic T cells, and natural killer cells. Okay, so gamma interferon therefore um, could have several functions. It's pleiotropic. So pleiotropy means that a certain cytokines would have several functions. Aside from gamma interferon, we also have the interleukin 2, also known as the T cell growth factors. So it drives the growth and differentiation of T and B cells, okay, and it induces lytic activity of natural killer cells. So therefore, we could say that interleukin 2 is important in cell-mediated immunity, also in humoral immune response. It also induced activity of natural killer cells, can activate also the proliferation of Th2. So interleukin 2 helps generate IgG1. This is the subclass of IgG and IgE producing cells. IgE producing cells, okay? So IgE will eventually be involved in allergic reaction. Okay, so that is the activity of the Th1 interleukin 2 cytokines. Interleukin 4, okay, as part of the a while ago, we're talking about the Th1 cytokines. This time, let's talk about the Th2 cytokines. So one of them is interleukin 4. So it helps drive antibody response in various diseases. So interleukin 4 could be involved in allergies, autoimmune diseases, and parasitic infections. So remember, we cannot destroy parasites using antibodies. Using the antibodies that are specific for viruses or bacteria, because parasites are actually much bigger. So in this case, we need a specific antibodies for them. Okay? So receptor expressed on lymphocytes and various non-hematopoietic cell types would also have interleukin 4. So another example of Th2 cytokines would be interleukin 10. So it has an anti-inflammatory and suppressive effects on Th1 cells. So interleukin 10 is actually produced by monocytes, macrophages, CD8 positive T cells. So yung CD8 positive T cells kasi, ito yung mga cytotoxic T cells. And then CD4 positive T cells, ito yung mga T helper cells. So interleukin 2 suppress the effects of Th1. Therefore, it has the ability to inhibit antigen presentation by macrophages and dendritic cells. And also, it serves an antagonist to gamma interferon to downregulate immune response. Mahalaga ba to? Somehow, yes. Because lalong-lalo na if we do not want to have graft rejection, or if we do not want to have an exaggerated response a certain pathogen, somehow it is important kasi nga anti-inflammatory. However, we do not want to have too much interleukin 10 because that will leave us defenseless against pathogen. So dapat level or balance lang yung production ng certain cytokines. Just plug in my power supply. Okay. T regulators or previously known as the T suppressor cells. So Tregs are CD4 positive and CD25 positive. So Tregs will help establish peripheral tolerance. Okay. What do you mean by the term tolerance? Meaning to say, no immune response will be mounted when we say tolerance to various self-antigen. This one is very important because we do not want to develop autoimmune disease. Allergens. This one is important, which means that we'll be free from allergy. And transplant antigen. This one is good because we reduce the possibility of graft rejection. What we do not want is the tolerance to tumor antigen and infectious agent. Okay, so it reduces transforming growth factor B, beta, that suppresses other T cells. So it can also induce interleukin 10 and transforming growth factor beta expression in adaptive TR1 cells in the peripheral circulation. So we just want to have a normal or balanced amount of Tregs. We do not want to have too many of them or too less of them or less of them. So T cell suppression occurs through interleukin 10 inhibition of pro-inflammatory cytokines. So if you will be asking me what cell will be would be anti-inflammatory or what cytokines would be anti-inflammatory, most probably it is interleukin 10. Okay? Because interleukin 10 is directly related to T cell suppression. So inhibition of co-stimulatory molecule expression on antigen-presenting cells. Okay, so the tumor growth factor beta can also downregulate the antigen-presenting cell function and blocks the proliferation of cytokine production by CD4 T cells. So if you notice, no, kung nasusundan ninyo yung lecture natin, cytokines can upregulate a certain scenario or event, or it can also downregulate a certain scenario or event. So in this case, Treg, whenever it is activated, the immune response is downregulated, and it is beneficial to us somehow because we prevent chronic inflammation. So we do not want our immune system to exaggeratedly react to a certain pathogen. Otherwise, we'll have a chronic inflammation. So let's talk about this time the TH17. So I did mention TH1, TH2, and Treg, but we haven't really discussed much about TH17. So TH17 are cytokines involved in both innate and adaptive immunity. So the TH17 cytokines have a role in host defense against bacterial and fungal infection at mucosal surfaces. Ano ba yung mga mucosal surfaces? This includes our eyes, our mouth. Okay, so these are examples of mucosal surfaces. So TH17 cells secrete interleukin 17 family of cytokines to produce to promote continuous recruitment of neutrophils. The TH17 cytokines will also promote antimicrobial peptides. When they are dysregulated, they are implicated in the pathogenesis of multiple inflammatory and autoimmune conditions. We do not want to have dysregulation. Kasi when you say dysregulation, this is the abnormal regulation or uncontrolled response. So uncontrolled response or abnormal regulation would result to inflammatory disease and autoimmune conditions. Okay, so we're done with the cytokines involved in innate and adaptive immunity. Let's talk about, this time, colony-stimulating factors. Perhaps the first time that you've heard about colony stimulating factors is when you were having hematology. Because colony stimulating factors are actually involved in hematopoiesis. Hematopoiesis, or the maturation and growth of different blood cells. So, colony stimulating factors include interleukin 3, erythropoietin or the EPO, the granulocyte CSF, the macrophage CSF, and the GM-CSF complex. So, this will stimulate the growth of hematopoietic cells in the bone marrow whenever whenever they are in response, or whenever they have responded to inflammatory cytokines such as the interleukin 1. So, if you still remember your hematology, the hematopoietic stem cell um, could be involved in various types of, it's actually the main cell wherein different cells of the body were derived. However, there are some specific cytokines that will allow growth and maturation of a certain cell. For example, um, by the way, the MP here, the cells from myelocyte progenitor, and these are the cells from the lymphocyte progenitor. Ito, mga lymphocytes yung napoproduce. Yung cells from myelocyte progenitor, divided into two major cytokines. The EPO will trigger the release of the RBC from the bone marrow. Do you know that EPO is produced by the kidneys? And then the GM-CSF, kay granulocyte myelocyte progenitor, will allow the growth and differentiation of basophil, eosinophil, neutrophils, and monocytes. Each one of them is being regulated by various types of cytokines. So let's talk about EPO. So EPO, or erythropoietin, is primarily produced by the kidneys. Do you know that during dialysis, patients are actually receiving EPO as well? Okay? So it regulates RBC production in the bone marrow. So RBC proliferates and induced by EPO and improves oxygenation. So whenever there is hypoxia or a low oxygen tension, EPO will be produced by the kidneys and it will trigger the release of the RBC in the bone marrow. Okay? So if oxygenation of the tissue improves, then the production of EPO will be switched off. Therefore, people with renal disorders will probably or have the possibility of getting anemia because of the absence of the EPO. And so that's the reason why it's very important. It's very important that you have a healthy kidneys to have a normal RBC level. Okay. So I will not go into details anymore. I just want you to remember that since this is not hematology, I just want you to remember that various WBC would have a corresponding cytokines. So for example, basophil stimulated aside from GM-CSF is also stimulated by interleukin 3, particularly during allergic reaction. Eosinophil is stimulated by interleukin 5 and granulocyte colony stimulating factor. Neutrophil is stimulated by granulocyte colony stimulating factor, and monocyte is stimulated by monocyte colony stimulating factor. Okay. So cytokines and anti-cytokine therapies there are various therapeutic approaches when it comes to cytokines. For example, there is a drug known as infliximab. Okay? So infliximab disrupt the interaction between cytokines and their cognate receptors. So meaning to say, um, infliximab somehow is actually anti-inflammatory or anti-cytokine. Okay? Some uses monoclonal antibodies, but monoclonal antibodies is cytokine agonist, meaning to say it will enhance the function of cytokines. Okay. So there is a drug known as etanercept. Okay. Etanercept will use hybrid proteins, and these proteins contain cytokine receptor binding sites attached to immunoglobulin constant region to block cytokine activity. So drug such as etanercept is also considered as anti-cytokine therapy. And some drug can even block the function of exekizum of interleukin 17, and these are the exekizumab. So, bakit binibigay yung mga anti-cytokine drugs? Anti-cytokine drugs are being given because somehow they are also anti-inflammatory. And and this somehow will also prevent cytokine storm. Okay. So how do we know the level of cytokines? So cytokines can actually be measured in our bloodstream through various methods. So for example, ELISpot assay will employ the ELISA technique on in vitro activated peripheral WBC. So ELISA spot or ELISpot assay. So we will talk more about ELISA. ELISA actually stands for enzyme linked immunosorbent assay during midterm. So there's another ELISA method called multiplex ELISA. The reason why they call it multiplex because it can detect about 12 to 25 pro and anti-inflammatory cytokines in one reaction. So that's why it's called multiplex. And the microbead assays, which allows for simultaneous detection of multiple cytokines in a single tube. So we will talk more about ELISA when we reach midterm. So that's about it. So that's the end of our discussion on cytokines.